Hello, everyone, and welcome to today's webinar, Can Definitive Drug Screening Become Routine? Challenges and Solutions. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Waters Corporation. Now, we encourage you to participate today by submitting any questions you may have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. Now, this webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain those credits. I'd now like to welcome our speaker, Dr. Thomas Rosano, a toxicologist and director at the National Toxicology Center and Professor Emeritus at the Albany Medical College. Dr. Rosano, welcome. You may now begin your presentation, sir.
Well, it's a pleasure to participate in this webinar session and to have an opportunity to share with you our experience in the conversion to direct-to-definitive drug testing program. In a prior presentation and webinar focused on the toxicology findings associated with this newer approach to drug screening across our toxicology practice. In this session, I'd like to focus on the challenges associated with really making that transition and also give you some sense for some of the solutions we found along the way in terms of addressing the issues of conversion. This next slide shows both the outline of the presentation as well as the listing of the challenges that we did face. The selection of panels, the technical issues, the informatic issues, and also I would like to address as part of this agenda the impact on workflow as well as economics. So let's start with the panel selection. I'm often asked what you may include when you are doing definitive screening, and really from a toxicology standpoint, that would be the agents that are on our streets and being used in therapeutics and other agents. They may be misused. One of the advantages we have for mass spectrometry is the ability to acquire data across many different analytes simultaneously, or with the rapid electronics associated with MS, the ability to acquire data for multi-analytes. And this is a listing of a panel of analytes that we acquire in one of our main panels that is always being acquired for all of the analytes. And then the selection of a panel that may be an agency's request for specific drugs or agents can be accomplished by processing only the data associated with the ordered drug tests. The next panel shows a panel that we used very early on, which in some ways mimics the class of drugs that were being tested by the presumptive panels, and included agents that were in those particular drug classes. And we used that for conversion of a number of agencies, but quickly found that, again, from a toxicology standpoint, we really need to go well beyond this, and as demonstrated in the first webinar, that we are missing a significant number of agents because the scope of this testing is limited. We use heavily a drug abuse panel that continues to evolve. This is a 70 analyte version of that panel that includes not only the classes of drugs that were covered by presumptive testing, but a more expanded class of drugs based upon the needs and the drugs that are being used and need to be identified. In addition, we have addressed a number of agents that are in the psychiatric practice that are also out and in use and may not be complied with. And in those particular cases, we've added antipsychotics, antidepressants, and this would be considered a comprehensive panel. So I should note at this point that the panel that each laboratory selects will be tailored to the needs of the various agencies that they're serving, and will be updated as the newer agents and designer agents continue to evolve in your region of the country and in your practice area. So the selection is such that we want to have acquisition of all the data. For example, we're now adding psilocin to that because we've had a significant number of cases of mushroom utilization and misutilization for hallucinogenic activity, and we do that by updating this panel over time. There are technical issues also associated with the transition to definitive drug screening, and I'm going to address two of them related to sample prep and related to the analysis by MS. Automation is a key to this. We have automated our preparation of samples. There are just too many samples in the initial testing process for us to actually handle in a manual basis. It's doable, but it is really not practical. This is an example of about 100 or so specimens, as well as calibrators, controls, and blind samples that are probably in those particular case samples. And they're being a liquid handler is being used for the preparation of those samples. We use exclusively 96-well plate approach and do the preparation in 96-well plate, the analysis in MS in 96-well plate format. And this is up in the upper left-hand corner, a 20 microliter sample being kicked up from each of the cases, as well as the controls and calibrators, and the next right-hand side shows the plates, the 96-well plates, where there's delivery of the samples, and then delivery of the reagents that are needed for that particular testing, and also other agents that dilutions and other reagents that are used are in larger volume and available for that rapid addition process. Down below, on the right-hand side, lower right-hand side, you can see that a plate is being transferred from the initial preparation area and brought over robotically to a mixing and incubation area. So it has temperature control. It has the ability for mixing of the components of that particular reaction mixture. And it then is brought back over for the final dilution. After that, it's hydrolysis and incubation, and then finally brought back for a mixing. All in an automated sense. Single plate is about 30 minutes for the preparation of the plate, and then depending upon your hydrolysis time, it will sit for hydrolysis. And then after that is automatically diluted and then available for analysis. So we'll pick the first plate off after it is fully prepared, while the second plate is being processed at the same time, and start our MS analysis. So in the center is the I-class acuity auto sampling system for the UPLC, and the use of that 96-well plate positioned within there. So automation is a key to the use of MS in a high volume setting. Some of the advantages associated with it are the elimination of manual additions. And to the extent that there are close to 300 additions that are needed for a single plate, this becomes a very both laborious process, as well as one that requires significant concentration and is potentially error prone in terms of manual additions. Whereas in the liquid handler, it is a script that is written that robotically does those same additions each time, can barcode identify samples, can barcode identify plates, and allows you, as well as other reagents, and allows you to have a much better control over the preparation of multi samples. The liquid handler script is prepped, and in our case is prepared for multiple plate preparation. There is a plate in which the hydrolysis, a plate submitted for preparation. The first plate can be done in sufficient time, so about an hour and a half into it, we are actually doing our MS analysis. The hydrolysis and the steps involved in the additions are on the lower left-hand side, and in that particular right-hand side, I've presented again the very important aspect of glucuronidation, the glucuronidase hydrolysis, in that a significant number of analytes would be missed because of low sensitivity, because they are in a glucuronidated form. So it's important that we actually go through that hydrolysis process. The second part of the analysis that's important to address in terms of high volume use is the chromatographic analysis itself. And we have developed on a high theoretical plate process using UPLC the ability to have a chromatographic separation in 3 minutes, using dual transition acquisition of data during that time frame. We focused heavily on diastereoisomers like pseudoephedrine and ephedrine separation, and have also focused on isobarics in terms of making sure that we have good chromatographic identity for that. But it's really this rapid separation that's important, again, in terms of processing that number of samples. Now, the use of this, once developed, and we developed this for urine testing program, has also now been adapted to our oral fluid drug screening program for definitive testing and screening initial oral fluid samples. And this shows the format for that, a little bit different in terms of the addition. We add some more. The sample volume is twice the amount as in the urine sample. Obviously the collection, we use a passive drool, a buffered oral fluid that was developed at NTC for that process, and a collection aid that is available and FDA cleared for collection of oral fluid. And so we've adapted this same approach to the use in oral fluid. Not only are the automated preparation of sample, the analysis and the by MS important, but also is how we do the analysis and the accuracy of that analysis. And I'd like to spend a couple of minutes on matrix effect and quantitation. Quantitation in the initial testing process, which is not normally done, but is easily handled in this particular approach. This is a look at the addition of 100 nanogram per ml concentration of MDA to 42 negative urine, analyte negative urine samples, and looking directly at the ion areas that are achieved with each of those samples. And you can see the wide range of ion area associated with an approximate 2,000 range up to 13,000 in terms of ion area. And that variation is essentially matrix effect. That is that the competition at the source for MDA ionization will vary from sample to sample depending upon other things that are also in the source. This is an example of the suppression, ion suppression associated with amphetamine by another drug. This is a sample that was initially, this is a panel of analytes, which were initially analyzed and achieved a baseline ion response, ion area. And then following that, there was the addition of more and more gabapentin to this particular sample, and then the reanalysis at higher and higher levels of gabapentin. Gabapentin co-elutes closely, has a retention time 1.31, as does amphetamine, and in that process is in the source at the same time. As you have more gabapentin, it is suppressing the ionization and the counts associated with amphetamine. That is a matrix effect caused by a co-analyte in that particular sample. So look at it in terms of an image that kind of tries to explain what is occurring. If you look on the left-hand side, you have a column in which you have, in this case, let's say a simulated sample, which is matrix-free. It is in your starting mobile phase, for example, as a sample, and it contains amphetamine. When that particular sample gets to the electrospray process and the ionization process at high voltage in positive mode, it produces positive ions of amphetamine. And there are a certain number of ions that are produced. They're not the entire number of molecules that are actually presented to it, but a significant or a portion of them are actually ionized and therefore taken up by, in this positive mode, by the MS source. If you add to this matrix another ionizable ion, in this case gabapentin, and have that also along with amphetamine, you can see at least a depiction of the fact that in this urine sample that now has amphetamine in it compared to a matrix-free sample, that because of this competitiveness, you're having ionization of gabapentin as well as amphetamine, and the net result is less amphetamine ionization is occurring, and that is why the ion response in that particular urine sample is different from the ion response in the matrix-free sample. The same is true from urine to urine, depending upon what is actually co-eluting and at the source results in that kind of MDA effect that I showed before in those 42 urine samples. It is therefore essential, I believe, in either qualitative work in terms of a threshold, using a threshold, quantitative work, that we have a way of normalizing the matrix set. And the primary technique for that normalization is standard isotope internal stable isotope internal standardization. And that has been used for a very long time and is widely used for quantitative work and confirmatory work for MS analysis. An alternate that we use in our laboratory that I pointed to in the last session is a threshold accurate calibration, a technique that is essentially like a single point standard addition, although it is different because it is, in fact, is calibrated, externally calibrated by the calibrants that are used in the process. I don't have time to go into it, but standard addition really would not work for a system in which the analyte itself could be competing as you put more and more analyte in. So standard is the typical standard addition that we used in toxicology for a long time does not work very effectively in electrospray MS analysis. But this technique is one approach. So the first is to have stable isotope standards. And we are fortunate that there are availability of stable isotopes for many of the agents. Often when a new designer agent comes into use, we are awaiting those internal standards for quantitative work. But this is an example of what I call analyte matched stable isotope standards. Each one of those analytes has a deuterated standard associated with it, or a C13 stable isotope analog that is used because it would be in the source at the same time, and therefore it undergoes the same level of enhancement or suppression that may occur for that particular analyte in that sample at that moment in time. The approach that we've taken was initially reported by us in 2016 as a novel calibration technique, and I'm just pointing to it as an alternate available method for this kind of matrix normalization, where we have a sample that has in it an analyte, and we take a aliquot of that and we add to it and spike it with a reference analyte, and you can see that you can have, therefore, the initial analysis, which is just the analyte, and then the spiked analysis, which includes both, in which you can do a quick subtraction of the two to come up with really the amount of analyte endogenous and the amount of added response for that particular sample. We call it the TAC ratio for that particular analysis. And you can see that as you take analyte matrix-free or two different samples that may have different matrices, that the amount of suppression that may occur for amphetamine, for example, is shown by the neat ion response for each of them. And you can see that they are quite different, and it would not be possible to calibrate unless you use the exact matrix for that particular sample. But by adding this addition and spiking process using this threshold accurate calibration, you actually get a ratioing process that normalizes so that you can calibrate with a matrix such as a single urine, negative urine, analyte negative urine that you're using, and can calibrate for all your samples and be accurate across the entire range of samples that you're analyzing at that particular run. We've also looked at this carefully in terms of the quantitative comparability to the stable isotope method, and this shows a correlation that we've reported recently in a publication that is in the special issue of JAT for this year, and is a comparison of various techniques. And in this paper is also a very important point that I'll make right now is that I've pointed out that you would use analyte matched stable isotopes. Many of us have not in the past used analyte matched stable isotopes where we've shared an isotope from an analog from another drug, for example. And I'll show you the issues associated with this, and it was reported, the head-to-head comparison was reported in this paper. And this is use of hydrocodone D6 along with for the measurement of hydrocodone for its identification and quantitation. And this is 7 different matrices, different urine samples in which we have been analyzing that. And you can see that the response is very parallel in terms of any matrix effect that may occur in each of these samples, because again, they're both in the source at the same time experiencing the same type of effects in terms of competition for ionization. However, if you look below and you try to use dihydrocodeine or codeine or some other analyte's internal standard to measure hydrocodone, there will be samples in which there is a mismatch in that in this particular case, the internal standard has been suppressed because it's moving through that source with a different set of ions and is not at the retained at the same time as the analyte hydrocodone, and it has a different effect in terms of ion response. And the net effect is when you add a certain level of drug and try to recover it, you recover very well that particular drug. I think it was at 230 or 235 nanograms, 235 nanograms per ml concentration. You find that when you have this kind of a circumstance in which you have a non-matched deuterated internal standard, you can have quite significant differences in the concentration. This won't be evident from your quality control and calibration, which was done in a single matrix, and that was pointed out also in that report. So there are two methods, but we should be very careful about analyte matched stable isotope utilization. I'd like to move on now to the management of the data, because it is one of the largest challenges associated with the use of definitive methods in the initial screening process. And I point that out by showing you the data volume that we are confronted with when we have these kinds of analysis of 100 to 200 samples. We have 800 transitions per case, and we have 100 to 200 cases. So we have thousands and thousands of transition ion counts that we are managing that data and doing that processing of results. In addition, we have to, because we are doing it in quantitative mode, we have calibration curves, 100 calibration curves, and in the range of 500 quality control per run. It is not possible to use some of the basic methods in terms of quantitation and calibration curves when you're doing this many calibration curves at the volume that we're using in this particular case. So what is our approach to it? Our approach is a data analysis, and this is really another challenge for the industry itself, is to be able to manage the data. But this is we ourselves export the data, the MSMS data, and put it into a template that already has all of the calculations, all the algorithms, all the criteria, and all the analysis rules, so that we can actually perform those. Those analyses are performed in an automated sense by just putting that exported data into the template. And it shows up, as I'll show you in a moment, all of our calibration curves. And it allows us also to review by exception for looking at anything that doesn't meet our rules or criteria, and have a much better managed approach to a volume of data that is quite significant in directed definitive testing. The calibrators and controls we find are best to look at from an analyte standpoint, so I'll show you that analysis and how the analyst and the certifying scientists look at that data. And the casework data is best to look at in terms of across all the analytes for that particular sample. So let me just give you the outputted calculations that are performed in this for a calibration curve. This is a norfentanyl calibration curve showing the calibrators and QC samples, showing the TAC. In this case, it was done by a TAC technique, so with a TAC ratio calculation for each of those samples. In this case, since it is TAC, we make sure that the recovery between the two injections is an acceptable criteria. So we have criteria around the recovery in which we put methapurolene into both samples, and then check for the accuracy of the 5 microliter volume injection into the mass spectrometer. We also have criteria for the quantitative recovery in terms of plus or minus 20% for calibrators and for QC material. The ion ratio criteria that is used in confirmatory method, we use in the initial testing method. The RT criteria in terms of the peak that is found, as well as this injection criteria or recovery. We also look at carryover and display that, and show you the limits of quantitation, the lower limits of quantitation, upper limit, as well as LOD. So sitting in front of the analyst for a look at quality control is all of the essentials, and we have them light up. We have an exception criteria. Well, they will not only say fail, but they will also light up, so you can very quickly see what problems you may be having with an analyte. Down below is the actual regression analysis for the standard curve, and a visualization of the standard curve, and an R-square for that particular standard curve. When it comes to the cases, once we are through our quality control for the 100+ analytes, and we quality control all the analytes, and therefore are ready to do any panels that are necessary for that particular run, we then look at our casework, and our casework also is driven by criteria of it, where we have the analyte, we have the RT for the calibrator, the RT for the analyte is then checked against that. The case TAC ratio, the calibrator TAC ratio for the one of the calibrators, so we can get ourselves in range on that, as well as the information of what the cutoff is for that particular sample, the lower limit of quantitation for the sample. Then we have criteria for the RT. If we do find peak for that particular sample. We look at the ion ratio for that, and it also has a criteria, and we also have an injection criteria that's used. And we also have a calculation that is done off of that calibration curve that I showed you before to give a quantitation for that sample. And the end result is, if we do find a sample that meets all those criteria, it will actually show up as a finding. So this is buprenorphine, norbuprenorphine. There's also benzoylecgonine found in the case, a gabapentin, naloxone, which is part of the buprenorphine therapy, oxymorphone, which is really nornaloxone. It is the identical in structure to it, and came from the naloxone metabolism. This is an individual that was on amitriptyline. You can see the amitriptyline level, the nortriptyline level, and they were also on trazodone and the trazodone metabolite, and they were also on a sedative, using a sedative agent, and they're smokers. So we also have cotinine. So there's a lot of information that comes from that particular test. So in the last area is to look at impact, and the impact on both our workflow as well as the economics. And to summarize, the workflow is on this slide, indicating that a single analytical platform is now used. We're not moving between immunoassay and the mass spec, between screen and confirm. We're on the mass spec for these agents on both the screening and the confirm. The automated sample preparation saves us significant time and gives us much greater quality assurance and reliability in the preparation of those samples at high volume. The ability for us to have a selected identification of drugs in their metabolites is now seen in that screening process. You just saw the results of an initial testing and the extent of findings that are associated with that. But it allows a definitive level of not only screening for positives, but also screening for negatives. So all the negatives are also being done and resulted via definitive method, and again, increases the certainty associated with those results. The batch analysis allows testing for up to 150 samples per run. The samples and the calibrators and the controls are analyzed, prepped, analyzed within about a 40-hour period, and then the samples are coming off as each chromatographic, each of them are analyzed in that MS process. And since we do such a significant number of samples, we will set them up and then overnight have all that analysis done so that we can then analyze the samples the next day and provide reporting. I haven't put a copy of our report, but I think it's important to know that just sending out the results of each of those samples or each of those tests that I showed you may not be sufficient. There's good information from Brigham's and Women's Hospital that looked at this, that found out that there's just too much information that might be put out, and various forensic agencies have just are not able to digest and really understand all of the information that's put out. So we put out interpretive reports that has key findings in terms of the drug finding and how that's associated with the tests themselves, and also allows interpretive information to be provided along with those reports. And finally, the economics. We recognize and know that the cost associated with the equipment is higher on the MS side for definitive testing, and that initial investment is higher than tabletop immunoassay systems that are out there, and therefore the direct cost of having that built into a direct cost will affect the relative cost of a sample in terms of the testing. And it is not until you actually have increased volume of testing that you ultimately have less cost associated with definitive testing. And that's because the variable cost associated with the two approaches is quite different between the reagent cost for presumptive testing and the ongoing cost per sample of definitive testing. And we find somewhere in the range of 10 to 15,000 cases, which is about 200 samples per week or so, is close to where we actually have a net savings from the use of definitive testing. So in summary, liquid handler coupled with a simplified sample prep, rapid chromatographic MS analysis allows the high volume throughput and solves those analytical aspects and workflow aspects in terms of testing. Matrix normalization is essential part of good practice in terms of having that using either analyte matched stable isotopes or other alternative approaches. Definitive screening data for calibrators, controls, and case is manageable. Casework is manageable by a rules-based, criteria-based, and review by exception. When you get into this kind of volume, that is the approach that is needed for handling that volume. And the cost effectiveness is really case volume dependent in terms of direct and indirect cost for it. So I answered to the question, and I posed this question as the topic for this webinar is, can definitive drug screening become routine? And our experience is that the answer is yes. And it does take a significant amount of solutions to challenges that I showed in this webinar. And I'd be happy to discuss those or answer any questions associated with our approach to it, and look forward to those discussions. So thank you very much for your attention.
And thank you very much, Dr. Rosano, for that informative presentation. We will now start the live Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So Dr. Rosano, it looks like we already have some great questions coming in from our audience. Regarding your test panels, how do you validate and added new drugs to the panels?
We're always in the process of adding and validating new analytes in addition to the panel. And the way we do that is after optimizing the conditions for each of those analytes in terms of optimum conditions of cone voltage, collision energies, retention time, we put that information into the acquisition program. So we're actually acquiring the data for those particular analytes while we're doing our runs. And we therefore have them in our calibrators, so we're calibrating. So we're getting all of our calibration curve data, we're getting all of our quality control data during those runs. We're not processing any case samples with it, but we are getting all of that data. So a great deal of the validation is actually performed during routine testing. And as I said, the number of agents we're currently working on now are being handled in just that way. They are being acquired, they are processed for quality control data, and allowing us to validate those particular analyses prior to use in practice.
Thank you so much. Now, did you include blind samples in your testing program?
Yes, part of the protocol for forensic laboratory and good protocol for other laboratories is the use of challenging us with a blind sample program. One of the things that we learned in this was because we added so many analytes that we wanted to make sure that we tested all analytes. So we have an interesting approach to the blind program where the certifying scientist is submitting samples blind to the analyst in which they include what would be considered to be reasonable relationships between drugs, metabolites, and various therapies that might be used or misused, drugs that might be misused. And those are then submitted along with the casework. So they're somewhere in that casework, and the certifying scientist will then evaluate that as to whether that is an acceptable run. So every run has it, and we run through, as I said, there's over 100 analytes. We make sure that we test through this blind program all analytes regularly, so we kind of rhythmically go through all the analytes in what is about a 14 or 15 different combination group that we send through as blind samples. So yes, we do use a blind sample program, very important process.
Thank you so much, Dr. Rosano. And I want to thank our audience for these great questions coming in. Dr. Rosano, what level of technical staff do you need in a direct to definitive drug testing protocol?
Well, in our setting, we need a licensed analyst, technologist to perform that, and they need to be trained in MS analysis and in the techniques and the approaches that we're taking in our laboratory to ensure the quality and effectiveness of those testing process. So it is not unlike other areas of specialty testing, not unlike postmortem toxicology, where the training of an individual, they may come with a significant amount of education, some experience, but the actual training is really on-the-job training that occurs. So we do need good qualified analysts for that testing process. And we also need certifying scientists to review the data that comes from that. And they are essentially analysts with the greatest amount of experience in our laboratory that are serving in that particular role, as well as the toxicologists.
Thank you so much. For our next question, an audience member wants to know, is your final report just a listing of screen and confirmed test results, or do you provide summary and interpretive information as well?
No, I thought of that during the session. I think I made a comment on it that I didn't show you an example of the report, but the report starts off with key findings. We do identify each of the tests that are positive, but we put it into a context. So initially, and the context initially is a summary context that is drug-oriented, drug use-oriented, so that we can be talking about the drugs and metabolites and other findings that may be associated with that particular drug use. So there's a key finding section. There is a initial test section, which is that screening process, and there is also confirmatory testing reported along with the quantitation. But if you look up in the top, you'll find interpretive information along with key findings that help to identify that. We never just list or alphabetize the tests or the test results. We think that really does not add to it. Having 100 agents just listed in terms of positive or negative really is not serving the interest of drug courts, probation departments, and others who are receiving or having testing performed for them.
Thank you so much. We have time for one more question. Dr. Rosano, do you have experience with using a limited number of stable isotopes for screening a large panel of drug and metabolite tests?
I think all of us in toxicology have, over time, really used that particular approach. And the experience that I best is a study experience where we actually, and it's reported in JADA, as I pointed to in the presentation, in which we actually took a look at using shared internal standards versus using analyte matched internal standards. And we found both the issues of recovery when we used multiple matrices, and we also showed the issue of dispersion of the quantitative relationship between the analyte matched approach and the shared internal standard approach. TAC and the analyte match are agreeing very well. In some cases, quite discrepant when the shared internal standard was in use. So we're just adding a caution that depending upon matrix effect, you may not, and the fact that your calibrators and controls may be prepared in a single matrix, that you are really not assessing effectively whether there may be a matrix effect in your individual sample. And so that approach is in use, but has to be used with a great deal of caution based upon reliability of accuracy.
Dr. Rosano, thank you so much for your presentation today. Would you like to provide any final comments to our audience before we go?
Yes, I think this two-webinar series was a good session for me also to kind of put in perspective a significant number of years of working through the issues and finally coming to a point at which we have it in routine practice and can show the value, the value in terms of the information that is provided in a drug testing program, and also hopefully advance the practice of drug testing so that it serves our agencies and others in a very effective manner. So it was helpful in the first session to look at the results of our finding, and I think even for myself it's helpful to look at and kind of analyze all the things that we had to do to try to get there. And each laboratory will do it differently with a different approach. And it's really this sharing of experience between laboratories that's probably most valuable. So I enjoyed it, and hopefully it has some benefit to others and other practitioners.
Well, we want to thank you again, Dr. Rosano, for your time and for your important research. And I also want to take the time to thank LabRoots and our sponsor, Waters Corporation, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted on the on-demand period will be addressed by the speaker via the contact information we provided at the time of registration. And this webcast can be viewed on demand. LabRoots will email you and alert you when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everyone, take care, be safe. Bye-bye.